Test for carbohydrate, proteins and fats. The food we eat is one of the necessary factors in our daily life that provides nutritional support for the human body. Food consists of both organic and inorganic substances. Carbohydrates, fats and proteins are the main organic substances present in food and provide energy. In this video, we learn how to test for the presence of carbohydrates, proteins and fats in food substances. Test for carbohydrates. One of the main components of our diet is carbohydrates. Glucose, sucrose and starch are all forms of carbohydrates found in our daily food. Let's start with testing for the presence of glucose in food substances. There are two tests, Benedict's and Fehling's test, used to find if glucose is present. Benedict's test. Materials required: banana extract in a test tube, Benedict's reagent, burner, test tube holder and a dropper. Procedure: using a dropper, take a small quantity of Benedict's reagent. Add the reagent to the test tube containing banana extract. Boil the sample over a burner for 2 minutes, holding the test tube firmly with the test tube holder. Keep shaking the test tube as it is being heated. A brick red precipitate appears, indicating the presence of glucose in the banana extract. Conclusion: on boiling banana extract with the Benedict's reagent, the cupric ion present in the Benedict's reagent is reduced by the reducing agent, sugar, to form a brick red colored precipitate of cuprous oxide. Fehling's test. Materials required: banana extract in a test tube holder, Fehling solution A, Fehling solution B, dropper and a burner. Procedure: using a dropper, take a small quantity of Fehling solution A. Add the Fehling solution A to the test tube containing banana extract. Using a dropper, take a small quantity of Fehling solution B. Add Fehling solution B to the test tube containing banana extract. Boil the sample over a burner for 2 minutes, holding the test tube firmly with the test tube holder. Keep shaking the test tube while heating. A brick red precipitate appears, indicating the presence of glucose in the banana extract. Conclusion: the cupric ion present in Fehling solution is reduced on boiling by the reducing substance sugar to form the brick red colored precipitate of cuprous oxide. Now, we can test the presence of sucrose in food substances. Materials required: sugarcane extract in a test tube, concentrated hydrochloric acid, sodium hydroxide solution, Benedict's reagent, test tube holder, dropper and a burner. Procedure: using a dropper, take a small quantity of concentrated hydrochloric acid. Add 2 to 3 drops of concentrated hydrochloric acid to the test tube containing sugarcane extract. Boil the sample over a burner for 2 minutes, holding the test tube firmly with the test tube holder. This hydrolyzes sucrose into glucose and fructose. Using a dropper, take a small quantity of sodium hydroxide solution. Add a few drops of sodium hydroxide solution to the test tube to make the solution alkaline. Now, we can perform Benedict's test to this solution to test for the presence of glucose. Using a dropper, take a small quantity of Benedict's reagent. Add the reagent to the test tube containing the sample. Boil the sample over a burner for 2 minutes, holding the test tube firmly with the test tube holder. The color changes from blue to green and finally to orange or brick red, indicating the presence of glucose. Let's test for the presence of starch in food substances. Materials required: potato extract in a test tube, iodine solution and a dropper. Procedure: using a dropper, take a small quantity of iodine solution. Add 5 drops of iodine solution to the test tube containing potato extract. Blue-black color indicates the presence of starch in the potato extract. Test for proteins. Proteins are large biological molecules made up of large number of amino acid units. The tests that are used to conduct for the presence of proteins in food substances are Biuret Test, Xanthoproteic Test and Millions Test. Biuret Test. Materials required: egg albumin in a test tube, 40% sodium hydroxide solution, 1% copper sulfate, test tube holder and a dropper. Procedure: using a dropper, take a small quantity of 40% sodium hydroxide solution. Add a few drops of sodium hydroxide solution to the test tube containing egg albumin. Using a dropper, take a small quantity of 1% copper sulfate solution. Add 2 to 3 drops of copper sulfate solution to the test tube containing egg albumin. Shake the solution to mix it well. A violet color appears in the test tube, which indicates the presence of proteins. Xanthoproteic Test. Materials required: egg albumin in a test tube, concentrated nitric acid, ammonia solution, test tube holder and a dropper. Procedure: using a dropper, take a small quantity of concentrated nitric acid. Add 5 drops of concentrated nitric acid to the test tube containing egg albumin. Boil the sample over the burner for 2 minutes, holding the test tube firmly with the test tube holder. Yellow precipitate appears in the test tube. Using a dropper, take a small quantity of ammonia solution. Add a few drops of ammonia solution to the sample. Shake the solution to mix it well. Yellow precipitate changes to orange in color, which indicates the presence of protein. Millions Test. Materials required: egg albumin in a test tube, Millions reagent and a dropper. Procedure: using a dropper, take a small quantity of Millions reagent. Add a few drops of Millions reagent to the test tube containing egg albumin. Wait for some time. Pink color appears in the test tube, which indicates the presence of protein. Test for fats. Fats are complex molecules made up of fatty acids and glycerol. They are found stored in many oil seeds and some animal tissues. The test that is used to conduct for the presence of fats in food substances is Sudan-3 test. Materials required: oil in a test tube, Sudan-3 solution and a dropper. Procedure: using a dropper, take a small quantity of Sudan-3 reagent. Add a few drops of Sudan-3 reagent to the test tube containing egg albumin. Shake the solution to mix it well. Pink droplets appear, indicating the presence of fat in the sample.